Harnessing microbes for industrial purposes using synthetic biology requires a systematic design, build, test, learn cycle approach, utilizing all the available tools on top of traditional lab work, from computational modeling to automation, robotics, analytics, and machine learning.
To achieve this, first we design the cell using computational tools to have a model of how to build the cell, and in our case usually a microbe, as we think it would work optimally. Then we build, that is, we engineer the cell according to the model using genome editing tools. Because with current knowledge our designs are not perfect, we need to test a large number of engineered strains, how they behave and what their cellular chemistry is, and finally we learn from the data to design improvements.
At VTT, the infrastructure and know-how in biotechnology and synthetic biology build on decades of experience and excellent in-house facilities from lab to pilot-scale testing. Before doing wet lab trials, synthetic biology offers tools for predicting the outcome of metabolic engineering of an organism. This includes a fascinating area of computational tools, analytics, and machine learning for engineering biology. At VTT, Paula Jouhten's team is specialized in this highly complex field of science.
Our team at VTT, we combine several kinds of expertise. We have computational expertise, so we do this data-driven modeling, and we do mechanistic modeling. So these tools help us to handle the complexity. So biology is dynamic, complex, these interactions that take place for some moments and then disappear. Biology is so highly complex that we could never ever cover everything just by exploring with high throughput experiments. For all this complexity we need help from both these mechanistic models as the metabolic models, but also these data-driven, so the machine learning models. And we believe that the combination of these models will be the success story in this field to develop learning cycles, and so that these computational approaches will be in the future efficient in suggesting the next modifications to cells and take into account the different kinds of functionalities in cells.
The modeling can significantly limit the number of wet lab trials as they are able to predict cell metabolism.
So these genome scale metabolic model simulations, they kind of give or take into account the ultimate limits of what is possible with the particular metabolism that the species has. So it is based on the ultimate laws of mass conservation and thermodynamics. So both of these become taken into account in the simulations. So we can handle with computational means in a predictive sense. So we can do predictions on what kind of chemistries the cells should be able to perform and how efficiently. So what should they contain? How efficiently should these reactions take place when they are metabolically active to meet our aims with the applications?
This method is especially valuable when venturing beyond the well-known model host organisms, such as the yeast Saccharomyces or bacterium E. coli.
For model organisms, there's a lot of accumulated knowledge, functional annotations, phenotypes recorded and such. But as we want to exploit the wide variety of natural species, we want also to be able to do this predictive modeling for other species, the non-model species. Many of them can be very interesting for applications. They have capabilities that the model organisms do not have. So what we can do is that all the information is there in their genomes. Genome sequencing is fast and affordable nowadays. So we generate these genome sequences. We functionally annotate the genome so that we can gather information of which enzymes are these species able to encode. So what biochemical reactions can take place in those cells. And then we assemble those into the network. So we do metabolic model reconstruction for any species from sequenced genomes.
But how do you derive new functionalities from the extensive sequencing data?
So there are huge sequence resources nowadays available, but we need to be able to there choose then the correct sequences that would have the functionality that we want to introduce into the cells of interest. And so for this reason, we need to understand how does it translate into functionalities. And so here we have done like conventional bioinformatics for tens of years already, and so this is efficient. So there's a lot of work put into also characterizing genes, and we use all this accumulated information on characterized genes and gene products, and we use this to identify new similar that could have similar functions but maybe different characteristics, maybe more efficient enzymes of the same kind, those ones that can convert the same substrates into products but more efficiently. So these are the enzymes that we look for in the sequence resources. But as we know, so there was a major breakthrough in the end of last year. So this AlphaFold 2. So you know the protein functionalities are highly dependent on their three-dimensional structure, and this whole folding problem has been a major problem or challenge in life science research. And so now there's a computational approach to predict the protein structures from the sequence. And this makes it possible then to use these structures that are the basis for the functionality. So we can use even these to identify novel sequences that could have the functionalities that we need for the applications.
Once the new cell functions are designed, the new genetic information, DNA, that is required for the desired functions needs to be constructed in a form that it can be inserted into the host organisms. So-called expression cassettes contain the genes and their regulatory functions.
Expression cassette is typically a promoter, then a gene and a terminator, which if you put it into a host, the promoter will harness the transcription, activation of the expression of this gene.
Once the vector is designed, the individual parts can be ordered online.
It has a huge options. It's a fully synthetic DNA, so you can actually order nowadays. There are companies which are actually making this fairly cheap, so you can order whatever DNA sequence you want, and they will deliver in a small tube DNA of your choice.
Once parts are assembled, there are several gene modification tools, such as CRISPR, to edit or insert the foreign genes into the host organism. At VTT, a fully synthetic expression system called SES has been developed for expressing genes in a huge variety of different eukaryotic hosts.
That expression system we developed a couple of years ago with the aim to actually be able to express genes in diverse organisms without need to use native promoters, native DNA parts of that particular organism.
The core of the system is that it is based on a universally active promoter element which enables the expression of all the subsequent parts of the system. So when the core promoter controls expression of the synthetic transcription factor, then any cell can express the transcription factor. This transcription factor then binds to DNA elements in a chromosome where we have engineered the binding sites for this transcription factor. The binding of this transcription factor to these binding sites then triggers the activation of the target gene that is under the control of the CES promoter.
This together forms the binding sites, and the core promoter forms a new synthetic promoter, new to nature. It doesn't exist in the nature, but it's functional in the fungi or in eukaryotic organisms.
And the important thing with the CES system is that we can not only express at very high level, but we can also tune the expression level. So, from very weak to gradually to very high, higher than the strongest endogenous promoter. In addition, our experiments have shown that the CES system is very stable and it maintains the expression levels also in bioreactor conditions. So, the set and designed expression levels are maintained throughout the bioprocess. An exciting feature of the CES system is that due to its modular structure, we've been able to use different features of the CES system to build complex genetic control circuits in cells. These circuits make use of DNA elements that enable either activation or deactivation of gene expression. Due to modularity, the CES system is well suited for high-throughput approaches using robotics. By using CRISPR technology together with CES modules, we can generate a large number of strain variants where the selected target genes are expressed at different levels. By combining these properties in different ways and targeting them to control key nodes in the metabolic network, we can direct cell metabolism for a designed end result. We have shown that it is possible to build genetic circuits that have a memory so that they enable cell metabolism to stay in a given mode even after removal of the short triggering signal.
Once the desired strains have been constructed, the automated testing facilities at VTT can be used, ranging from microtiter to well-controlled bioreactors of different sizes.
For the test phase, we have various automation platforms where we can test the strains in various formats. So we have the screening robot system where we can test the strains in a microtiter plate format. Then we have a biolector, which is a bit larger volume, and there also that's in the a bit larger microtiter plate. And then we have an Amber 250 system, which is then a bioprocess automation system where we have 24 bioreactors, and there we can also test the strains in a larger scale, which then resembles traditional bioreactors, which then also represent the pilot scale and the industrial scale that we aim to do.
We can carry out benchtop bioreactor experiments, so those are typically ranging from 1 liter up to 5 liters. We have standalone reactors, 10, 20, 50 liter scale, and then also the piloting equipment, and that's ranging from 50 liters to 1,000. And then we have quite well equipped downstream processing equipment system downstairs, so we can do cell separations, filtrations, up concentrating of broths, and various types of downstream processing steps there.
When scaling up, what kind of challenges are met when dealing with larger volumes?
Especially when we talk about process scale-up, there are several things we need to take into account when the reactor size is increasing, like mixing is a typical issue in the process scale-up, and aeration. Microbes often they require a lot of oxygen input, and that can be challenging when you scale up the process. And then we have different types of side streams we are working with, so pre-treatment steps before you can actually ferment your microbes. Then we think about other like purification related costs, so that's often like something that you don't really need to think about that much when you do experiments in small scale. But then in our pilot facility, then these are the things that we really need to see that the workflow works well. And for that, at VTT, we have quite unique platform to carry out all those different steps.
The analytics team at VTT have decades of solid experience and can carry out a multitude of measurements.
We have the bioanalytics and small molecular analytics in the team. So we can do fast measurements, we can do extensive measurements, we can do targeted analytics that are suited for certain applications. We develop novel methods there.
After testing, the DBTL cycle continues to analyze the experiments and incorporate what has been learned into the next cycle. And the basis for the learn phase is all the gathered data.
The high throughput power, that's a very cool thing that we can have so many individual experiments simultaneously, and we can gather so much data out of one day of work. And then if we go to the larger scales, and then we can measure various things from the same experiment. So for example, in the Amber system, so we can measure all the things that you can measure from a traditional bioreactor, and we can also take the samples automatically. So we can have a sample every hour for let's say 7 days. So we can also there gather a large pool of data that we can then utilize in the DBTL cycle.
We are really looking forward to see like fast DBTL cycle, because this allows us to accumulate the data so that finally we can employ so efficient machine learning algorithms to guide this engineering process. So some of these approaches are already possible. So we can apply machine learning to help the scientists by suggesting new modifications to the next round.
So Paula, what do you think are the expected benefits of the interplay of human scientists and machine learning tools?
Yeah, I think we'll become faster. Will become faster because by the scientists looking into the data and thinking, so we are clever, but we are slower. So if we can combine, like maybe even this combined kind of human guided machine learning, that sometimes the scientist can interfere and take part in the learning cycle, but that the machine learning algorithms will enable us to do this faster, and this will be of course then supported by the automated means for engineering cells.
And if you think about the big picture, what does it truly entail?
I think we should understand evolution better. So the possibilities of the evolution, and it has several kinds of effects on biotechnological applications. So on one hand, we would like to have our engineered functionalities to become stable, that they would function there, that they would not get changed by selection and by other evolutionary forces. And then on the other hand, evolution has enabled formation of all these super exciting characteristics that we see in nature. So could we actually use this better? So if we say that we could take into use all the functionalities that we have seen in nature, but what if we could actually use these forces to develop something new? Something that we can use for the applications. So I think this will be a big theme in future, like synthetic evolution.